Okay, so welcome everybody to our BioRender webinar on design tips for graphical abstracts. And for those that are brand new to BioRender, here is a sneak peek into the platform. It's a beautiful drag and drop interface. We do have a PDB plugin, as you see here, for those that use the PDB. But really, it's a beautiful drag and drop interface with tons and tons, tens of thousands actually, of vector-based icons, ready for you to use for your images. And again, today's focus will be graphical abstracts. Common use cases include publication figures, presentations, lab meetings, grant applications, and the list goes on and on. So hopefully you'll find some use for BioRender in your day-to-day and would love to see what you create. All right, so what is a graphical abstract? Let's start with the really simple definition so that we're all sort of on the same page about what a graphical abstract is. I think we all cover very different fields of life science and beyond. So the examples I'll use in this webinar, they will skew a little heavy towards cell biology. So apologies if it's not exactly relevant to your work, but again, the tips I'm covering will be very ubiquitous, totally applicable to any type of science figure you're making. So a graphical abstract in our simple terms is a single image that is intended to give your reader or editor, I suppose, if you're still in the submission process, an immediate understanding of the story or article's main message. So your graphical abstract should actually be distinct from figures or diagrams in the rest of the article itself, i.e. it should be an overview as opposed to one of those, panel figures in the results section. So I actually interviewed a cell press editor prior to this, just to get a sense of, what are common mistakes they see when they receive submissions for a graphical abstract. I know cell press has made the use of graphical abstracts very popular. I know they value a lot of graphics for ability to use to highlight an article, perhaps to use in social media to be able to advertise or really give a snapshot of what the article is about. So it's great. The advice was that the number of words used to describe the graphical abstract should actually be less than the number of words in the, that should say abstract itself. So, actually, you know what? I'm going to go ahead and change that because I'm a little picky here with the design. But suffice it to say that if you go the reverse order, where you actually describe your graphical abstract in words, it should actually be less than the abstract. And I know that abstracts usually have a word limit, say 300 words or 500 words, whatever it is. Graphical abstracts do not have a word limit, nor does it have a content limit. And that's where we get into a little bit of trouble here, because we'll tend to cram in everything that we weren't able to say in the abstract into the picture. And that's actually what this editor specifically found, is that a lot of authors found it as an opportunity to cram in information that otherwise would not have fit in the word limit. So it actually should be the opposite. It should be a lighter version of the abstract, perhaps take 70% of what was said and depict it in illustration form. And just as a refresher, I did a really quick Google search here. Hopefully you can still see my screen, of a publication figure. Just Googled it, very simple. And I was looking, and it's clearly, as you all know, a little more data focused, less, I guess, pictorial or cartoon-like. And I would say alignment and spacing tends to be the important part in creating better panel figures for publication figures. If you type in graphical abstract now, look at the variety of the types of figures, the amount of content and color, variety, the organic structures that get introduced. Sometimes it's more of a snapshot, an overview of the background of the science itself, which definitely leads to a little bit more nuance and creativity, and also some design mishaps, because not everybody went to school for medical illustration, which is totally expected, but unfortunately it's also expected of all of you to create beautiful graphical abstracts. So we're here today to give you some sort of tips to better make these more colorful graphical images. Okay. So back to my slide deck here. This was a great example of a figure that we helped create with a researcher here at the University of Toronto. This was the before image. So created in PowerPoint. Many of you have been here before. And it was actually a great foundation because all the content was there. There's some pretty decent hierarchy of text, directionality here with large arrows. But here's the image we made in just 10 minutes in BioRender, a little bit more sort of alignment and comparison in sort of a left to right format, a hierarchy of text, limited color palette, which is always important to keep in mind. And actually, we're going to do a live demo of how we compose this, just to show you how easy it is to take your before image that you might be sort of grappling with now into a cleaner after image. And actually, this image was certainly published. Actually, this is in Immunity, and we were able to help out the author to create a cover piece. So not only did we go from a figure that had a lot to be desired as far as design into what became the cover piece for this issue, which was a great sort of success story in our mind. All right, so this is sort of the general, I guess, categories that we're going to follow. It's actually a little different from this, but I wanted to kind of clump it into 4 general categories. And without further ado, let's dive right in. Just going to take a quick gander over at our poll that we launched at the beginning of this. Looks like we're doing okay. I'm going to end the poll. Sorry, I didn't get everyone's votes in there. But it looks like we've got a nice divide between previous BioRender experts and perhaps people brand new to BioRender. So that's great. All right, so for those that of you that know, this is the BioRender interface. We've got my figures here in the bottom. So these are just images that I've created or I've created from a template, or perhaps somebody has created and shared with me. You can actually share figures amongst yourselves, your colleagues, and your friends. I've got folders on the left-hand panel here. I like to stay organized by project, or maybe it's by manuscript or by presentation. And then if you navigate up here to BioRender templates, you'll see that we've got, I think, over 1,000 now pre-made figures that you can sort of tear apart, change the colors, edit the text. They're all sort of pre-baked templates, or pre-baked figures, I should say, that you can go ahead and revise and make your own. So if I type in something like cancer here into the search bar, you'll see that we've got beautiful cancer-related figures. And you can actually just use this straight up as part of your graphical abstract, saving you a lot of time in the process. Okay. So that was just a very quick overview of BioRender. This is not going to be an extensive, how-to on the BioRender app. We do have those webinars. Actually, we ran one today at 12 p.m. Eastern. We do them every Wednesday, so you're welcome to attend that, and we'll go through, how to use BioRender from beginning to end, the more nitty-gritties of it. Today, our focus, of course, is on graphical abstracts specifically. So I've created slide decks within BioRender. So I'm going to go through a couple of those and then go through a tutorial on how I would actually build out a figure. I think it's important to go through some of these basic concepts, review them for those that maybe have heard of these once before, and then really ingrain it into our mind so that when you go to make your own figure, you're not having to refer back. Eventually, it'll just come as second nature. So the first thing I like to highlight is thinking about layout and story flow. So this is the first of our 4 major topics here. Layout and story flow is really important. We always recommend sketching it on paper, whether that's the back of an envelope or a napkin or a lined piece of paper, just something to get your thoughts down. And we find that, pencil to paper, there really is no replacement for that. Sometimes you can use, a clean BioRender canvas and throw on a bunch of icons and maybe drag it around to get a general sense of composition. If you're good with Photoshop or Illustrator, you can also use that. But I find starting on paper to be the best start. And you've seen this before, likely, if you've attended a previous webinar, but we love to follow one or two of these simple compositions. It's really hard to follow the composition of a figure, the content of a figure, if things are sort of flying around left and right, up and down to up sometimes, or right to left. So keep this in mind. You don't have to memorize these, but generally speaking, it should fall left to right, sort of, directionality that we read, or up to down, the direction of gravity. Just naturally, we like to read up to down. Okay. So the first tip here regarding layout and story flow is sort of treating our thought process or creating a diagram to something as silly as this. When we were kids, you probably played the spot the difference game, where you had to sort of divert your eyes from left to right and look at the differences between the two images. We find that that's actually pretty analogous, pretty similar to the way scientific figures are composed, especially when you have sort of a disease state and a normal state, or a normal state and maybe a control and a variable. There tends to be a lot of comparisons, visual comparisons at that. So when you do compose diagrams, we highly recommend making sure that things are generally aligned and similar in horizontal alignment. So for example, this before and after here of a gene turned off and turned on. I'm going to create some alignment grid lines here in BioRender. You can see here that the proteins are almost perfectly aligned left to right when we're looking at the comparisons. So on the left, it looks like it's non-phosphorylated. On the right, it is. If these proteins, if I remove these guidelines for a minute, if these proteins were perhaps a little bit lower down or off to the side like this, it actually wouldn't read as well, because my eye now has a little bit of trouble comparing left and right. I don't know what really is the difference, what has changed. I can kind of see it if I really strain my eye and read closely, but it's really a lot friendlier for your audience if you are doing a comparison of normal and abnormal, that those are exactly aligned left to right or up to down. Okay, so don't do what's happening here. Make sure that they are aligned like so. Okay, this one is a really good example that I love to show at every webinar. This was bravely volunteered by a researcher, and they were trying to put together a graphical abstract. And the content is there. You've probably been in a situation where you feel like, your figure feels cluttered and you don't know how to fix it. A lot of the time, what's happened is that the reading order or the composition has gone a little bit astray here. So if I were to take this BioRender marker and follow the numerical steps here, I'm going to start up at number 1 and look for number 2, come up to number 3, 4, and then, the arrows also speak very loudly. There's no words on the arrows, but those also speak very loudly. So make that a different color here so you can see. Look at, if I were to be tracking someone's eye around this figure, how disjointed it is. It clearly doesn't follow one of the rules that we had mentioned here. And try and see if this is maybe what's causing the confusion in your figure, if the reading order is not consistent or clean. So basically what we did, I won't go into details for this specific figure this time, but we kind of flipped things 90 degrees. We moved and nudged around a couple of the elements. And this is what we ended up with. Here's the after, here's the before, here's the after. And sometimes it's frustrating because in design, there's no right or wrong answer. It actually takes a little bit of jigging around and nudging things left and right. But I'm going to do the same thing here. I'm going to start number 1, 2, 3, and 4. And you can see I also sort of followed the general arrow directionality, which was a little bit more like this. So much smoother composition. It's almost the exact same content. We just nudged stuff around, created a hierarchy of text. We created a hierarchy of arrows. That's also something that we don't see enough is you can vary the arrow thickness as we did here and really commit to maybe 1 or 2 strong, I guess, gravitational pulls for the composition itself. All the little arrows are kind of like side roads that are sort of side stories, but having a really strong directional arrow like this helps to lead your viewer's eye around the composition. So we went from, again, this to this. And this one does indeed follow our cyclical composition rule. Okay. So really simple overview there of layout and story flow. Kind of glossed over some of the details, but just keep in mind, again, this chart as we go through some of the other examples. All right. Second topic is color, one of my favorite topics. I can probably talk about color and the proper use of color for hours and hours. This is a very small clip and an example of, I guess, what not to do in your graphical abstracts. Again, I'm skewing a little heavy towards the cell bio fields, but you know, hopefully you can take away some of the general rules here. The first rule being color association. So whether you like it or not, if you start to color your diagram so that some elements match in color, your audience is going to automatically start to create associations simply because they are colored the same. Even if you didn't intend to. For example, these 3 cells are sort of a purple hue. These 2 are sort of a greenish hue. So in my mind, I'm thinking maybe these 2 cells are more closely associated than these 2 cells, even though that may not be the case. So sometimes we run out of colors to use, but do keep that in mind that you know, I would be careful with your color choice. For example, if I decided to color this B cell red for whatever reason, my eye would probably then associate it with this RBC right away, even though it really has no association in this composition at least. Okay, so just be careful about color association. Generally speaking, get away with it for H&E staining. So if everything was sort of a pinkish, bluish, purplish hue, people would probably assume it's okay. But just keep that in mind. Perhaps vary your colors a little bit if it ends up being that you can't get away from needing to use colors. Along that same vein, color and shapes do get associated very closely. For example, if these phosphates were to have been colored something like this, at a glance, your viewer might assume that those phosphates are associated with these little molecules here, which they are not. So you have to be careful about the shape and the color that you're using throughout your composition because they could be associated. Sometimes even these numerical little circles, if they're colored something fancy, like a blue, see how immediately that gets a little bit confused with this NF kappa B. So you want to be careful again about using colors that are too close together and using shapes that are too similar. When in doubt, go with as simple a color as possible for something like text. I would just go straight up white and black, like so. And then if you're using something like this, like a color like red, for something else in your composition, make sure you vary it greatly if it's similar in shape. I'll stick with maybe a yellow here. Okay, so they might seem pretty obvious, but just wanted to point that out so that you're not sort of starting off on the wrong foot already just by using the wrong colors and shapes. And really great question I'm seeing in the chats here. What about the size and text of in the figure? So we'll get into that, but that's absolutely another thing you have to be careful of. Here, I think we're doing an okay job, but generally speaking, you actually want to vary the text size and the style. Sometimes if you have, 1, 2, 3, 4, 5, these should all probably be bolded to look a little different from the label text here. You see the difference? Because these aren't necessarily labels. This is very different from, say, this body of text. Those are very different. They're communicating different things. So that's a great point. All right. Another thing is to really limit your color palette. This is something that I probably see quite often, and that's the desire to fill your image with color. In this case, we've limited the color palette to pretty much just white, a little bit of black for text, and then green being the fluorescing cells here throughout the whole composition so that nothing else is distracting and the story carries through from numbers 1 to 6, even in the graph itself. Okay. For example, if your mouse was something more colorful, maybe if it was a black mouse, that's okay too, actually, because it would still stand out. Basically, the point is, I wouldn't come in and start to make the text all colorful, something like, a red or a blue. That just starts to add unnecessary layers and complications, other dimensions into your diagram that is not necessary. So use color very sparingly is a take-home message here. Actually, even in this case, we've decreased the opacity of the organs and of the mice because we really only want the metastatic areas to be bright green. So this mouse used to be 100% opacity, but we knocked it back just to be able to see the tumors a little bit closer. Do you see that difference? Yeah, don't be afraid to play with that opacity slider. Super helpful. One little silly tip I like to always highlight here is to understand when to use cool colors versus warm colors on the topic of colors. So here's an example of a pretty good color palette. The leukocytes, white blood cells are all the sort of cool color, and this cancer cell is reddish hue. That's usually generally what we like to pick. I see this, generally everyone doing a pretty good job of this, but I do sometimes get to see the opposite where the good guys or the protagonists of the story are colored these sort of warm colors. And it gets a little confusing as to, what is damaging to the body and what's actually helping. So just to keep things aligned, stick with warm colors for harmful things in the body, whether it's, bacterial infections or cancers or virions or pathogens. Make those all a warm color. Also, we like to color inflammation to be reddish as well, so that makes sense. And then again, something like a blue or a green, a cooler tone for all the sort of good guys in the story. Okay. Second to last point here on color, really important. Probably the number one mistake we see in graphical abstracts, and that is contrast. So you hear us talk about this a lot in other webinars, but it's definitely worth repeating here. We often see these sort of like middle grayish or middle toned colors on top of middle toned colors. And that's where we get into trouble because those foreground elements or the items on top, layered on top, they tend to disappear because they're too close in color value. That's what value means up here. The darkness or lightness of that color is too similar to the thing that it's layered on top of. And what happens is it totally disappears. Say if you're colorblind, if you had to print this in black and white, if you had a poor quality monitor at home, perhaps, So a nice gut check is to use our preview in grayscale mode in BioRender, as you can see here. It'll knock out all of the sort of medium toned colors. I'm going to turn that back on. And it'll tell you right away the problem areas of your figure. So you can see here the DNA does look like it disappeared, or at least half of the strands disappeared. Same with the cells. So if I were to go back here and choose a darker color for the cell, you'll notice that right away it pops. See that difference? So just make sure that you're doing that final gut check, or maybe midway gut check, using our grayscale checker. This right here, view canvas and grayscale button to see where you've got contrast issues in your figure. Another way to combat that contrast problem is to, again, use our opacity slider. So in this case, the main point of the story is to really highlight the growth of the tumor in the lungs. If I use my trusty grayscale button, you'll notice that it doesn't really pop out to me. I'm really distracted by the trachea, the lungs, the liver. There actually is a tumor in the liver, but you probably didn't see it because it's sort of a similar shade or value. So when I turn off this grayscale mode, I'm going to come back and shift select all of the lungs, maybe even the liver, and come up here to the opacity slider. And you might be scared to use this because you might think it'll lose that contrast, but you'll be surprised how light you can go without losing legibility. And really, you know, basic scientists or anyone with like a undergrad level understanding of science should know the general shape of a lung. I think this is pretty symbolic. So they don't really need to know the exact outlines of it. Same with the trachea. If I were to come up here and maybe reduce the opacity, look how much the lymph nodes pop out, how much more clear it is. I mean, you could probably go way down to like 25% opacity and still get your point across, actually even more. I probably wouldn't go that light, but you can see here the before and after is pretty stark. There we go. So the point here is don't be afraid to use different opacities when you're sort of layering objects like this to make the point of your story really stand out. All right, let's go to the next topic here. Arrows and lines. There's just one thing I want to highlight here in the different types of arrows and lines we have. We have this great worksheet that I sometimes go through in more detail. In this case, I'm just going to focus on how to label your diagram better, because this is going to save you a lot of clutter in your next diagram, hopefully. So what I'm going to do is, instead of using this simple arrow, which I usually see when people label their diagrams like so, this is probably what you're used to doing, is just grabbing a label with an arrow and then just pointing to the thing. And it'll do the job, but what will do the job even better is to replace this arrow with a dot. And actually in BioRender, you don't have to even delete this arrow. You can just come up here and change the arrow type to this dot. You'll see here the difference. And then I would probably even bend this at the middle like so. See how much cleaner of a result that gives you? And also doesn't give you this sort of dead spider look with the lines going in all directions. You can actually use a line that is actually parallel to the word that it's labeling. And all you have to do, and by the way, I'm alt dragging to duplicate objects on the canvas. There we go. So keep that in mind, using this sort of hinged line with a dot at the end when you're creating a label for your diagram. And if you want to review, this is available in our worksheets category, in our templates category. 6 different ways to create lines in BioRender, which I won't go through today, but just keep in mind this label type line. And then lastly, looking at alignment and spacing as a theme. And then I'm going to go into a bit of a workshop on how to create a diagram with some sketching and importing into BioRender. I'm going to go back to this concept of that spot the difference game and now apply it again to alignment and spacing, similar to composition actually. But wanted to show a couple of diagrams. This is available as a template in BioRender. So actually a lot of templates in BioRender would be appropriate as parts or whole graphical abstracts. So if you're short for time and you find the exact image you need, just give it a little bit of an edit so that it's unique to you. And you can actually go ahead and use that as a graphical abstract. Looking here at what this template is trying to convey, this figure is trying to convey. It's nice that it's colorful. The left side is blue, the right side is yellow. But what it's communicating to me with those color differences is that we're showing two different cell types, when that's not the case. Actually, what's showing here is just two different ways in which synapses are formed in a neuron or between neurons. So really, this should be the same color as the elements in the right. So I'm going to quickly update that. There we go. Actually, maybe it's more of that orange hue. Yeah, there we go. So exactly the same left to right. And same thing below. I'm going to create a color combo that's a little bit more similar left and right. Now, this is one of those unique shapes like this. It's not a brush or an icon. So what I'm going to do is select both, come up here to fill, and then color pick. And I'm going to color pick this exact color. There we go. It looks like this shape is a brush. So I'm going to come up here and try to pick a brush color that's similar to what's shown on the right. I think that's pretty close. This one looks good. Okay, so it's one of those, again, successful spot the difference games. Everything looks generally the same left to right, but now the differences are really popping out. You know, I'm seeing vesicles on the right where I'm not seeing it on the left. The blue cell color is not a distraction anymore because everything's the same color. The channels are different colors, so that's telling me that you probably have different functions. The ions are sort of shown in this sort of the cleft here or the space, whereas here it actually doesn't enter the space because there are these connections that directly channel those ions through. So you can see here how much more successful the message is here because I've kept it consistent left to right. Same thing with this image. Could have been relatively complex if, say, the capillary was on the left and these cells were on the right. It's a lot friendlier on the viewer's eye, the fact that everything is aligned left to right and only the differences are highlighted. So perhaps the color of this epithelia, is changed a little bit. By the way, this is a really cool brush we have in BioRender that you can use to manipulate complex shapes like this. So this being the enterocytes in the gut. Let's see here, showing a normal capillary versus an inflamed. It's in the exact same position, left to right. Even the mast cell, if this mast cell was, up here somewhere, it might look like, a different cell has entered the scene, but really we're showing the same cell either activated or inactivated. Okay. So those are, again, sort of using left to right comparisons when you're showing normal or abnormal states or steps in a process. Another thing with alignment and spacing, this is just, sort of traditional alignment and spacing tip. And that is to use the align function in BioRender to align some of the labels. So something like this to make sure that they're all horizontally aligned. But also don't ignore the sort of vertical alignment that makes figures that much cleaner. So if this is step 1, 2, and 3, actually, let's add that in really quick here. Step 1. By the way, how I got that was coming up here to insert shape, text shape, and clicking these little pre-made circles, which is great. I'm going to alt drag so I can make a number 2 here. And zoom in so you can see. Alt drag and create number 3. So sometimes what I see is that we ignore vertical alignment while the horizontal alignment is pretty well and good. So what I like to do is create some grid lines. You can come up here to the ruler, place down a couple of grid lines just to be able to align your diagram a little bit better. Sometimes I'll throw down some horizontal lines as well. But as I always say, if you go into a professional design studio, you'll probably see everyone's monitors covered, littered in these guidelines. Just something that designers notoriously love to use because they don't trust their eye. And if they're available, why not use it? So this is 4. I'm going to double click to edit these numbers, and 6. Great. And you can hide the grid lines if it looks noisy and you can't see the rest of your figure. I'm going to hide them temporarily. Oops. And I can bring them back by saying show all guidelines. But I'm going to hide them for now. All right, what else can I do to clean this up? Maybe I'll shrink down this mouse a little bit, shrink down this microscope. You can always shrink down the things in your diagram that probably don't need that much real estate. And already we've got white space opened up in my diagram. All right, let's do a really quick tutorial here on how we came up with this graphical abstract. And then we're going to wrap up with a really simple tutorial on how to collaborate on a diagram. Okay. Really quick here. Hopefully you can still see my screen. I'm going to dive into Photoshop. I know I'm cheating here by using Photoshop, but I needed to use it because you can't all see my desktop if I were to sketch on paper. So what I usually do when I go about doing a figure is I look at sometimes what's already available. So this might be an old figure that you have lying around that you want to fix up. So here it is. Here's that diagram that I showed earlier. Okay, and that's what we're going to learn to fix up here. Sometimes I have the abstract itself handy just so I can see and refer back to, again, the content. And I have here the simple rules of composition. This might be on a different monitor or something like that, or I can print it out. What I'm going to do is look at the composition here and see what is sort of wrong with it. Look for any repeating elements that you could align side by side or stack on top of each other, like you showed, that sort of, spot the difference game. Check if there are any clear arrows or unnecessary arrows, if you can remove those, or arrow types. Are there too many types of arrows? Zoom in. Let's see. Are there clear or obvious themes? So there's clearly, a hierarchy of text here, signal 1, 2, 3, 4, maybe some smaller labels throughout, things like that. Where are areas that we can eliminate color? Is it too colorful? Can we just knock it back to black and white and then start from scratch? Sometimes I even do that, is I just take an image and I make it black and white, like so. Okay. Let's see. And then, what are some simple compositions that we could test out, looking again at the compositions down here? Finally, you do want to look at your journal specs. So if we come here, I'll just grab, yeah, this is good. Sometimes we'll have to just trace out, roughly, I think even cell press specifically is a 5:5 ratio that you have to stay within. So sometimes you'll need to draw out a very rough outline of the shape that you need to stay within or dimension. Okay, let's try out a couple of scenarios. I know that there's never going to be a perfect science to this, but sometimes you'll have to just test it out. So looking at this composition, maybe I'll do something like, signal on the left like that, and draw a couple of arrows. Maybe there's a dendritic cell and then some sort of T cell. It could be really as messy as this. And sometimes I'll take another color and just show maybe where the labels would go. And I'm already feeling like there might be too much going on left to right here. I might start to lose some space. There's going to be another arrow coming this way. Let's try more of a vertical composition. So something like signal being up here, the dots. Let's see. Maybe it's going up to down. This composition is going left to right. Let's try a composition going up to down. So let's try dendritic cell 1 and 2. Again, very messy, totally non-committal, just sketching it out. And then maybe this does work because now that I have a top-down composition, I have a little bit more room left to right for some labels. So this could work a little better. Okay, so if you're kind of happy with your very rough sketch, what I sometimes do is export that. I'm gonna do a quick shortcut here and just screenshot right into my composition. All right, zoom out and paste. All right, so what we did here was we took this sketch and we composed this on the right with some TV magic here. We're not going to go into the details, but what we did was we kind of searched up some of these simple icons in BioRender. So if I were to type in dendritic cell, something like a beautiful dendritic cell would come up. These are actually templates. I'm going to navigate over to icons. And we've got some really neat dendritic cells here you can select from. And we've got T cells if you were to type in a T cell. So pretty self-explanatory. All the ingredients are here, ready for you to just place and recompose. But a lot of the work ends up being in just figuring out the general composition and then sort of sticking with that layout as you create it. Okay. As a final step here, as we sort of wrap up our tutorial, as I promised, I did want to show a really quick tutorial on how you could probably collaborate with a colleague. I suspect that many of you do have to create figures together. And in BioRender, there's a brand new option to be able to share a diagram in real time and be able to edit it at the same time. Right now, you can only share a copy, but what you can do with our new real-time collaboration feature. So if you come up here to share, I'm going to add in my colleagues that are in the call now. Got Allison, John, and Cindy. There we go. I've added those 3 individuals. Right now, I can't change permissions. It's just edit. Send invite. And you're going to see them show up here in the top right as if it was one of those Google Docs. And we're going to be able to share and collaborate in real time and probably save me a lot of time in making this graphical abstract. Oh, here we go. They're showing up. And soon you're going to start to see them editing the file, moving stuff around. And what we should show, actually, maybe what we will do is paste a link to signing up for this really neat real-time collaboration feature. So you can see here, John has selected this protein. Allison is coming in here, making edits to this text. Really neat way to be able to collaborate together. Instead of, I'm sure you've all done this, attaching a PowerPoint file to an email and then going back and forth on version 1, 2, 3. This is a really great way to be able to speed up your illustration process. I've also have uploaded here a image I exported from GraphPad Prism. So you can do that. You can actually import any figure that's in the form of a PNG, a JPEG, or PDF. And I've added that there to my diagram to finish it off. And by the way, that's in this upload tab if you ever need. Okay. So maybe what we'll do is we'll paste the link to join the waitlist in the chat. And then what you can all do is if you don't already have access to real-time collaboration, actually, maybe you do, maybe a handful of you have already tried it. You can go ahead and sign up for that. And let's see, why don't we pause there and wrap up? I did want to remind you all that graphical abstracts do serve the purpose of highlighting and summarizing your diagram for those that are reading the actual journal, but it also serves a purpose on social media. For example, this tweet here, it actually takes up the majority of the tweet. The graphical abstract itself becomes sort of the preview or the trailer for the article itself. So it will get a lot of use case if done right. And it'll serve as sort of the cover piece for your story. All right, so that was a lot of content. I hope you were able to take in some of it. Again, this is pre-recorded, or it's recorded, so you can actually watch the recording again later. We'd love if you would try out the real-time collaboration feature. It is new. I would love your feedback on it as well. And we've linked it in the chat there, so you can actually click on that link, sign up for the waitlist, and then you'll usually get taken off the waitlist within a week. And do keep in mind that if you are making a figure for graphical abstracts in BioRender, you do need a premium subscription for at least a month. And we have a free trial, a 14-day free trial that you can try BioRender Premium, but you do need to have at least 1 month subscription. You could ask your department head, as we do have a lot of site-wide institutional licenses with companies and schools all over the world. So you may already have access and you may not know it. And then finally, one last ask is that we have a free swag giveaway. We always love doing this at the end of every webinar. Lots of swag to give away. So if you could give a little bit of feedback on a few questions. One is, what is one pain point that you are experiencing in figure making? And then, if we could help build one feature in BioRender, what would that be? We'd love your insights there. So that's it from me. Thank you so much for joining, everybody. Had a really huge crowd, a lot of engagement, tons of great questions. Thank you so much to John, Cindy, and Allison for answering all of your questions as well. I think they deserve a round of applause. They did an excellent job in keeping up with all of your questions. And as I said, we ask for these 3 things here. If you could provide feedback, try out real-time collaboration, sign up for the waitlist, and then try the free trial for BioRender Premium. It'll give you all the bells and whistles in BioRender, and there's no risk there. You can try it. You can always downgrade risk-free. Great. I think that's everything for this session. We've got a lot more content coming up in the rest of the Design Tips webinar, so we hope to see you there. And we'll sign off here. Thank you, everybody, for joining.